Today we're going to be stepping into one of the most powerful stories in all of biology, the story of how life changes through the time and where it began. Evolution gives us a scientific framework to understand everything from the structure of a finch's beak to the rise of humans. As we move through this presentation, I want you to think about evolution as the process that connects every living thing on Earth. It's not just history, it's the ongoing story of life. When we talk about evolution, we are talking about the changes that happen across generations within populations. These changes take time. No individual evolves. Populations evolve as their traits shift across many generations. These differences come from the genetic variations that already exist or from new mutations that appear randomly. Evolution is also shaped by unpredictable forces like the environment or chance events. As we move forward, keep in mind that evolution is both slow and constant, working quietly over millions of years. The fossil record is our window into deep time. It's incomplete because soft tissues rarely fossilize, but the fossils we do have give us the powerful timeline of ancient life. By looking at rock layers and using dating techniques, we can place fossils in order and understand how life form changes. When we combine fossils with the genetic and anatomical evidence we have today, we get a clearer picture of how evolution unfolds. Another source of evidence comes from comparing anatomy and developmental across species. When two organisms share structures with slightly different functions, they usually point to a shared ancestor. On the other hand, when unrelated organisms have similar structures because they live in similar environments, that tells us something different. Embryos also add clues because early development stages often reveal deep evolutionary connections. These comparisons help us trace the path that life takes. Homologous structures gives us some of the clearest evidence for evolution from a common ancestor. When we look at the forelimbs of humans, dogs, whales, and birds, the function could not be more different. Humans use their arms for grasping and manipulating objects. Dogs use their forelimbs for running. Whales use their flippers for powerful swimming, and birds use their wings for flight. Even with differences, the underlying bone pattern is the same. Each limb has a humerus, radius, ulna, carpals, metacarpals, and phalanges arranged in the same basic layout. These similarities show us that all four species inherited the structures from a shared ancestor, and over time, each lineage modified it to fit its own way of living. It's a reminder that evolution often works by reshaping what we already have. Analogous structures can look very similar, but they do not come from a shared ancestor. Instead, they evolved independently when different species face similar environmental challenges. A classic example is the wings of a bird and the wings of an insect. Both allow these animals to fly, yet the structures are built in completely different ways. Bird wings are modified forelimbs with bones, muscles, and feathers, while insects are thin extensions of their exoskeleton with no bones at all. Their similarities come from convergent evolution, where nature arrived at the same solution through different paths. This helps us see how powerful evolution and environmental pressures can be in shaping form and function across unrelated groups. Vestigial structures are fascinating because they show us the physical traces of our evolutionary history. These are structures that once had important functions in ancestral species but have lost most of their original purpose over time. They remain not because they are needed, but because evolution modified existing structures rather than removing them entirely. We see clear examples outside of the human. Whales still carry small pelvic bones deep inside their bodies. These bones once supported hind limbs when their ancestors walked on land. Snakes, especially pythons and boas, have tiny pelvic bones that are remnants of their legged reptile ancestors. These structures do not have functions the way that they once did, but they still hold valuable evolutionary clues. Humans have vestigial structures as well. The appendix is a reduced version of a larger cecum used by herbivorous ancestors to digest plant materials. The tailbone or coccyx is that remains of a functional tail. Tonsils are smaller remnants of an early immune structure system. Wisdom teeth reflect a time when our ancestors had larger jaws and needed extra molars to grind tough plant material. Together, these examples show us our bodies carry the imprints of traits that were once used but changed as the environment and lifestyles evolved. When we compare embryos, we see similarities that can be hidden in adults. Vertebrate embryos show features like pharyngeal slits, tails, and repeated body segments. These early stages similarities tells us the different vertebrate groups shared a deep evolutionary relationship. As development continues, each lineage modified these structures in a different way. Embryology is a reminder that evolution leaves cues not just in bones, but in the development programs written in our DNA. Evolution also leaves its mark on the world map. When we study where organisms lived and how they are distributed, we start to see patterns tied to geography and history. Mountains and oceans and deserts act as barriers that separate populations and allows them to evolve independently. Continental drift adds another layer. When Pangaea broke apart, species that were once neighbors were suddenly carried to different parts of the world. Over millions of years, these separations created entirely new lineages. Biogeography gives us real world examples of how isolation drives changes. Thinking about Earth as a constantly shifting system helps us understand why certain species appear where they do. Millions of years ago, all continents formed one large landmass called Pangaea. As it began to break apart, it changed the evolutionary trajectories of countless species. What we see today is the distribution of plants and animals in a reflection of those slow geological movements. Understanding continental drift helps us connect Earth's physical challenges and changes with the biological changes that followed. Evolution is not driven by a single mechanism. Instead, it is shaped by the combination of forces. Mutations create new materials for variation. Natural selection filters that variation and favors traits that improve survival and reproduction. Migration adds or removes genetic information from populations. Random events can shift gene frequencies by chance. When we combine all of these processes, we start to see evolution as a dynamic and ongoing interaction between genes, times, and environments. This sets us up perfectly for the next slides where we look more closely at natural selection and genetic change. Charles Darwin is central to our modern understanding of evolution, not because he was the first to notice changes in nature, but because he explained the mechanisms behind it during his voyage on the HMS Beagle. He collected observations from around the world, including the unique species of the Galapagos Island. These observations shaped his insight on species changes over time through natural selection. When he published On the Origin of Species in 1859, he provided the foundation that still guides biology today. The finches on the Galapagos Islands are one of the clearest examples of natural selection in action. Each island had different food sources, and over generations, fish populations evolved beaks that matched their local diets. Thick, powerful beaks helped crack seeds. Long, slender beaks allowed birds to eat insects. This diversity came from a single ancestral species. The finch show us how one lineage can branch into many when the environment favors different traits in the different species. Natural selection works through a few simple but powerful steps. Individuals in a population vary. They compete for resources, and not all will survive. Those with traits that help them in their environment are more likely to live long enough to reproduce. Over generations, these traits become more common. Natural selection is not purposeful. It does not plan ahead. It simply favors what works in the present condition. This process can slowly reshape a population in remarkable ways. At the heart of evolution is genetic variation, and the source of new variations is mutations. Mutations are random changes in DNA. Most are silent or neutral, but some alter traits in a meaningful way. When a mutation is not harmful, it can be passed down to future generations. As these small changes accumulate, they give populations a wide range of characteristics. Without mutations, evolution could not have raw materials to work with. Variation is the fuel that natural selection acts upon. Natural selection shifts the genetic makeup of populations over time. Traits that increase survival or reproduction tend to spread, while harmful traits become less common. What counts as harmful or helpful depends entirely on the environment. A trait that is beneficial in one situation might be disadvantageous in another. This link between genes and environment is why evolution never stops. As condition changes, the gene pool adjusts. This sets the stage for the roles of genetic drift and gene flow, which we'll explore next. Evolution can also occur through random changes, and that is where genetic drift comes in. Unlike natural selection, drift is not based on whether a trait is helpful or harmful. Instead, chance events shift which allele becomes more or less common. One example is the bottleneck effect. When a large population suddenly shrinks due to a catastrophe, the surviving group may not represent the original genetic diversity. This can change the trajectory of the population's evolution in ways that have nothing to do with adaptation. Another form of drift is the founder's effect. When a small group leaves to start a new population, the allele frequencies they carry become the foundation for the new group, even if those frequencies were rare in the original population. In contrast, gene flow works by moving alleles into or out of a population as individuals migrate. This mixing of genes can increase diversity and reduces differences between populations. Drift and flow reminds us that evolution is shaped not only by selection but also by the movement of randomness that occurs in real ecosystems. When we look at the history of life, we see that evolution has not been a steady, gentle process. There has been dramatic movements when huge numbers of species disappear in a short period of geological time. Then there's mass extinctions. They open new ecological space and reset ecosystems. After each extinction, surviving species diversify to fill the empty niches. These events have shaped major transitions in life on Earth, including the rise of mammals and eventually humans. A mass extinction happens when at least 75% of the species disappears within a relatively short time frame. These events are usually triggered by a major disruption to the environment. Some are caused by rapid climate change, some by asteroid impacts, and others by massive volcanic eruptions. Even though these events are devastating, they are also followed by periods of evolutionary creativity. Life rebounds and diversifies in new ways, revealing how resilient the dynamics the biosphere can be. The first major extinction we know of occurred roughly 443 million years ago. Most life at that time lived in the ocean, so when global cooling caused sea levels to fall, marine habitats changed rapidly. Around 85% of marine species disappeared. This event likely sparked the glaciation and chemical shifts in the ocean, clearing ecological space for new marine lineages to evolve. Even in deep time, climate shifts played a powerful role in shaping life's extinction. The Devonian extinction happened around 360 million years ago, and unlike a single catastrophic event, it unfolded over a long stretch of time. Marine ecosystems were hit with the hardest. Coral reefs collapsed and many fishes groups disappeared. Scientists think low oxygen conditions in the ocean played a major role, along with volcanic activity. There is also an interesting connection to the rise of the early land plants. As plants spread across land, they changed soil chemistry and nutrient runoffs, which may have disrupted ocean life. This extinction shows how changes on land can ripple into the ocean. The Permian-Triassic extinction is often called the Great Dying because nothing else compared to its scale. About 252 million years ago, more than 90% of marine species and around 70% of land species went extinct. Massive volcanic eruptions in Siberia released huge amounts of carbon dioxide, which led to rapid warming, ocean acidification, and cascades of environmental stress. When we talk about major turning points in the history of life, this is the one of the biggest. It cleared the way for new groups to rise, including the earliest relatives of the dinosaurs. After the Great Dying, life slowly recovered, only to face another challenge about 201 million years ago. The Triassic-Jurassic extinction opened the door for the dinosaurs to dominate. The cause again points to major volcanic activities, this time from the Central Atlantic Magnetic Province. These eruptions released tremendous amounts of carbon dioxide, which drove climate change and ocean acidification. Many early reptiles and amphibians did not survive the environmental shifts, but the group that did were able to diversify in and newly opened spaces. This is the most famous extinction by far because it ended the age of the dinosaurs. The Cretaceous-Paleogene extinction, which was roughly 66 million years ago, was caused by a large asteroid that struck what is now the Yucatan Peninsula in Mexico. The impact triggered global fires, tsunamis, and months of darkness as dust and debris blocked sunlight. Plants struggled, food chains collapsed. The groups that survived, including the ancestors of modern mammals and birds. Without this event, the path to human evolution would have looked very different. We are now arriving at the extinction event unfolding in our own time. The Anthropocene extinction, also known as the sixth mass extinction. Many scientists argue that human activity has pushed Earth into its sixth mass extinction. Unlike previous extinctions caused by volcanic eruptions or asteroid impacts, this one is linked to habitat loss, pollution, climate change, overexploitation of species, and the spread of invasive organisms. Extinction rates today are far higher than the natural background rate. At the same time, conservation efforts show that human actions can also protect and restore biodiversity. Understanding these patterns of loss helps us see responsibilities in shaping the future of life on Earth. Evolutionary trees or phylogenetic trees helps us visualize how species are related through time. Each branch point represents a divergence, the moment where one lineage splits into two. These trees are built using data from fossils, anatomy, and modern genetics. When a branch ends before reaching the present, the species is extinct. Sometimes we see bursts of branching called adaptive radiation, where many new species appear in a short lifespan. These trees help us trace the history and story of life as a series of branching points, not a straight line leading to humans. Now we shift from the evolution of species to the origin of life itself. Early Earth was nothing like the planet we know. It formed roughly 4.6 billion years ago and was far too hot to support life. The atmosphere was filled with gases like carbon dioxide, methane, ammonia, nitrogen, and water vapor. There was no oxygen and no ozone layer, so the surface was exposed to intense ultraviolet radiation. As Earth cooled, water vapor condensed to form the first oceans. These warm, shallow seas became the setting where the earliest chemical reactions of life began. The earliest evidence of life dates back to roughly 3.8 billion years ago and possibly even earlier. The first organisms were simple, single-celled, and lived without oxygen. Life began in environments that today would seem extreme. Over billions of years, evolutionary processes transformed these early cells into a vast diversity we see now. The long span of time between the first cells and the first rise of complex life reminds us how gradual and patient evolution can be. One of the biggest questions in biology is how the building blocks of life formed. The Urey-Miller experiment in 1953 provides an important clue. They recreated the gases of early Earth in a closed chamber and added heat and electrical sparks to simulate lightning. After running the system, they found amino acids and other organic molecules formed spontaneously. This showed us that under the right conditions, simple molecules can assemble into ingredients needed for life. It does not explain everything, but it gives us a starting point for understanding how chemistry became biology. Once organic material existed, the next step was assembling them into the structures that could store information and replicate. Some scientists proposed that early self-replicating RNA formed on clay surfaces or near volcanic heat. Eventually, these molecules became enclosed in a simple membrane made up of lipids or proteins, forming the earliest cells. These cells were anaerobic and relied on chemical energy instead of oxygen. As photosynthesis evolved, oxygen began to accumulate, transforming Earth's environment and paving the way for more complex life forms. Photosynthesis marks one of the biggest turning points in Earth's history. About 2.7 million years ago, certain prokaryotes developed the ability to capture sunlight and use it to produce energy. The key byproduct of this process was oxygen. At first, oxygen was toxic to many early organisms, but over time it accumulated in the atmosphere. This shift changed the chemistry of the ocean and paved the way for organisms that could use oxygen for efficient energy production. The rise of oxygen allowed life to become more complex and sets the stage for evolution of eukaryotic cells. Prokaryotes were the only life forms on Earth for nearly 2 billion years. During that time, they transformed the planet's atmosphere and laid the groundwork for more complex cells. Around 1.8 billion years ago, eukaryotic cells appeared. These cells had an internal membrane and a nucleus to protect their DNA. The endosymbiotic theory helps explain how this happened. Larger cells engulfed smaller prokaryotes, and instead of digesting them, they formed a partnership. These internal symbionts eventually became mitochondria and chloroplasts. Once eukaryotic cells existed, evolution had the tools to build more complex organisms. About 1.4 billion years ago, the first multicellular eukaryote appeared. These early forms were simple, but they allowed for specialization, where different cells would perform different tasks. Over hundreds of millions of years, multicellularity led the rise of larger and more diverse organisms. Eventually, animals, plants, and fungi emerged. This period represents one of the major transitions in the history of life. When we put everything together, we can see the long arc of life's history. Earth formed roughly 4.6 billion years ago. The first prokaryotes appeared 3.8 billion years ago. Photosynthesis began about 2.6 billion years ago. Eukaryotes around 1.8 billion years ago, followed by multicellular organisms about 1.4. Life in the ocean became more complex, and around 450 million years ago, plants and animals began to move onto land. The timeline continues through the evolution of vertebrates, animals, primates, and eventually humans. Human history occupies the smallest sliver of the entire story. To understand where humans fit in the tree of life, we use taxonomic classification. Humans belong to the domain Eukarya, the kingdom Animalia, and phylum Chordata. We are mammals in the class Mammalia and primates in the order Primata. Within the family Hominidae, our genus is Homo and our species is Homo sapien. A species is different and defined as a group of organisms that can be interbreed and produce fertile offspring under natural conditions. This classification helps us understand how evolutionary relationships with other animals. Humans are part of the larger story within the mammal and primate groups. As mammals, we share traits like hair, specialized teeth, and mammary glands that produce milk. Within the primates, we share flat nails, forward-facing eyes, and flexible hands to help with grasping. These features reflect our evolutionary past and the environments our ancestors adapted to. When we compare ourselves with other primates, we see both our similarities and the traits that make our lineage unique. When we trace primate evolution, we follow our branching paths to the spans of roughly 60 million years. Early primates give rise to lemurs, monkeys, apes, and eventually humans. Each branch represents a group that adapted to different environments. For example, New World monkeys evolved in the Americas, while Old World monkeys remained in Africa and Asia. The hominoids branch, which includes apes and humans, shows the emergence of traits like bigger brains and more complex social behaviors. Hominids are the great apes, which include orangutans, gorillas, chimpanzees, bonobos, and humans. Within this group, hominids refer specifically to modern humans and species more closely related to humans than to chimpanzees. Hominins share traits like bipedalism and increased brain size. Chimpanzees and bonobos remained our closest living relatives, which means our evolutionary path split from about 6.7 million years ago. Studying hominids helps us understand the steps that led from early apes-like ancestors to modern humans. Human evolution is marked by several major trends. Bipedalism allowed early hominids to walk upright, freeing the hands for tools use and changing the anatomy of the pelvis, spine, and our feet. Our brains gradually increased in size, supporting language, culture, and complex thought. Facial structures flattened, and our teeth became smaller as diets changed. Hands became more dexterous, which supports tool making and technology. These trends did not happen all at once. They unfolded over millions of years and shaped the biology and behavior of our species. The fossil record gives us a timeline of hominid evolution. Around 6 to 7 million years ago, species like the Sahelanthropus appeared in Africa. By 4.4 million years ago, Ardipithecus showed us a mix of traits for climbing and walking. Between 2 to 4 million years ago, groups like Australopithecus and Parthenopus appeared, while the earliest members of the genus Homo began to emerge. These fossils helps us piece together how anatomy, behavior, and environment interacts through human evolution. Each discovery adds another layer to the history. The transition from Australopithecines to the earliest members of the Homo genus is still one of the biggest questions in human evolution. We know that about 2.4 million years ago, Homo habilis appeared in the fossil record. These early humans showed evidence of tool use, which marks an important behavioral shift. The lack of clear transition fossils reminds us that the understanding of humans' evolution is still incomplete. New discoveries continue to shape what sometimes challenge our interpretation. Homo erectus represents a major milestone. This species appeared roughly 1.9 million years ago and was the first hominin to leave Africa and spread into Europe, Asia, and Indonesia. They had larger brains, larger periods of childhood development, and more advanced tools. Their long presence on Earth, nearly 1.8 million years, shows how successfully they were at a species. Their ability to adapt to new environments sets the stage for later human migration. For much of human history, we were not alone. Several Homo species lived at the same time. Homo heidelbergensis spread across Europe, Asia, and Africa. Homo neanderthalensis lived in Europe and parts of Asia and showed complex behaviors such as caring for injured individuals and creating art. Homo florensis lived in Indonesia and had a much smaller body size. Homo ladalli is another recent discovery that was an interesting mix of ancient and modern traits. The Devonians are also known mostly through DNA, showing their evolutionary past is still richer than what we had once. Modern humans originated in Africa between 300 and 160 million years ago. We see increases in brain size, more refined tools, and evidence of language and culture. Between 70,000 and 50,000 years ago, humans migrated out of Africa and eventually spread across the globe. As they moved, they encountered and sometimes interbred with one another Homo species, which is why modern humans carry small percentages of Neanderthal and Denisonian DNA. Today, Homo sapiens is the only surviving member of the genus, but our genome is still reflected in the deep history of these interactions. When we follow this entire journey from the first organic molecules to the rise of modern humans, we see how evolution shapes everything in biology. The story of life is not a straight line, but a branching network of adaptations, extinctions, and innovations. Studying evolution helps us understand how species change, how ecosystems respond to environmental pressures, and how our species came to be. It also reminds us that life continues to evolve and we are part of the ongoing process.